I'm Pejman Mohammadi. I'm a faculty at Scripps Research in La Jolla, California, in San Diego. And so today I'll be talking about a lot of stuff within the genomics of transcriptome analysis, basically. So one of the perks of being of the last talks in a conference is that you kind of watch people chipping away at your stuff. So I thought actually I'll take this opportunity to basically go over some simple, not so sexy stuff that are kind of interesting if you actually want to do these things. And so there are a bunch of nitty-gritty details of and facts and things that would be helpful if you're trying to do some of the analysis that we're talking about today. It's going to be a little logically scattered, but just bear with me. I hope everyone, or at least a decent amount of you, can stay awake by the end of it. Okay. Oh, and by the way, throughout the talk, there's a ton of works cited in there. Majority of them are not my works, but this is a tutorial, so I'm not going to one by one point them out. So, all right. So why do we work on regulatory genome? So this is a figure from a paper that came out not long ago, basically showing the growth of genomics knowledge. So on this side you have the common trait disease associations that basically we kind of didn't know anything about until we started to get good amount of data to be able to do GWAS, and basically it started taking off right after that pretty quickly. And so here is this 2018 with 80,000 associations. That's has wildly changed by now. I think I looked up GWAS catalog the other day. It's already at around 300,000, and that's from about a year ago update. So this thing is exploding down there. And then on this end you have the Mendelian diseases. So this is rare diseases, monogenic, single variant causing the whole thing, that we kind of started picking up on them way earlier because they're just in some sense easier to do. And now we know a few thousand of these genes that are causing these sort of diseases. So that's really cool. And the big problem that still remains is that on both sides of this spectrum of diseases, we have a big problem that for majority of the cases, we still cannot fully understand what's going on. So on the GWAS side, majority of the stuff fall outside of the coding genome, and basically as soon as they're out, we are for the most part lost as long as they're not in some sort of smoking gun region in the genome that you exactly know what it does. And for the most part, we don't. And then on the other side, you have the rare diseases that Val talked about them in pretty long details, basically. But majority of the patients that come to clinic with clear genetic disorders, you actually cannot diagnose them at the molecular level when you do whole genome sequencing of the entire family, because basically, as soon as you don't have a clear-cut, loss of function, a smoking gun gene of your interest, basically you're kind of lost, and then there are too many things to look at. So anyways, so a lot of people are looking at regulatory genome to basically try to make sense of some of these cases. And so briefly, so when we're talking about regulation, so there's two main parts of it. There's cis and trans. So I'm going to just introduce it for the purpose of this talk so that we know. So the cis-regulation is going to be the cases like this, where you have some sort of regulatory variant sitting somewhere on your basically one of your haplotypes, and it affects the regulation of the genes that are physically sitting on the same molecule, and that contrasts to this case of trans effects, where you have some sort of genetic variation somewhere in your genome, whether this case or that case, and then there is some sort of diffusible mediator. So for example, maybe there's a protein, or maybe there's a microRNA, something like that, that basically goes somewhere else in the cell and then change the regulation of some other gene. But this is not having an allelic effect. So the effect is shared between both alleles. I kind of feel like I should have attached this to my chin instead of this. But at any rate, so now in practice, usually cis and trans are used the same way with close, like proximal and distal. And there is truth to that. So most of the cis effects are by definition close in the genome, but obviously the genome is not sitting like a straight line. So there are definitely cis effects that are further away, but in practice, all we know is usually pretty close by. And the same goes with the trans in the opposite direction. So for the purpose of this talk, I'm going to talk only about cis effects. So things that affect one allele in the genome at a time, and they don't care about the genotype in the other one. And so why do we? So what are the kind of cases that you could use to study of regulatory genome? So there are so many things, and you could do your own, but I can point out some of the things that we're interested in my lab. So this part would be basically using regulatory phenotypes or transcriptional phenotypes or any other sort of intermediate phenotype you could get your hands on as some sort of like a stepping stone. So that's something that Sasha talked about it in detail yesterday, so I'm not going to go through it. Basically co-localization, TWAS, all sorts of good things. And they basically help you interpret GWAS variants, hopefully. And then on the opposite side of the spectrum of this would be using regulatory variation to identify when you have a rare variant of likely large effect in your genome. So I'm not going to go into the details, but for example, this is like an example plot from a paper we had in the rare variant subgroup of GTEx with Stephen Montgomery in Alexis Ball's lab. But basically, we looked at a few transcriptional phenotypes you could readily extract from your RNA-seq data and looked at individuals that seemed like outliers with respect to the rest of the like the data in the GTEx data. And so here you could see, so up there you have outliers of gene expression for up and down regulated, and then AAC outliers and splicing outliers. Doesn't matter what they're the details of it, but basically the color parts are the rare variants that we could associate with different kinds of genomic events, whether it's duplication or there are structural variants like deletions or change of splice sites or mutations in conserved non-coding regions. So you could actually pick up different signatures, and as you could see, like one thing you could see easily from this pretty poor quality plot that I put, which is probably not justified given my age, but like it's like these colors are different. So basically different kinds of outliers actually give you information on different kinds of mutations that are happening. So, and then finally, this sketch is from a paper from Tully Lepeleni's lab with Stefan a few years ago. That what they did there was that basically they showed that if you have some sort of large effect coding mutation, so think some sort of loss of function coding mutation you have in a gene, depending on common regulatory variants that you can have on that haplotype, basically the regulatory landscape of that haplotype, you will end up with more or less of the good and the bad protein of that gene, and that actually can be traced into the phenotype. So you could, in this case, they were showing it in the case of breast cancer and autism, that you could see that basically people that are in the disease cohort are more likely to have the bad allele with higher expression, and vice versa for the healthy people. So basically, like that's another part of studying regulation, is that putting the rest of the variants in the genome into their regulatory context that they actually are. So okay, so how do we map regulatory variation in populations? So this is so cis-EQTL calling. This is the most established way to identify basically parts of the genome that are predictive of gene regulation. And it's a very straightforward idea. So basically, if you have a gene that you're interested in, you pick some sort of window that you can afford to do. Usually it's 1 megabase or less. And then you pick every variant, each variant at a time. You separate your population into 3 groups based on their genotype at that variant. And then you fit a linear regression there and basically see if this is significant. So this is basically like any sort of association study just done in the locally usually. And then basically if you could find that, you call it a cis-EQTL. Now this is not like necessarily a cis signal, but generally when you do this sort of proximal EQTLs, there's a large majority of them that are having actually a cis signal. And so there have been a lot of consortiums doing that. GTEx has is probably the largest and of the the most recent ones. I'm not going to go into the details. Several people already talked about it. But so just briefly describing for those of you who are not familiar with GTEx. So GTEx is a data set of generally healthy people, post-mortem samples that were collected at transplant grade, and in total basically includes about 1,000 people, around 20,000 RNA samples that are collected from about 50 tissues. It's not some sort of rectangular design, so some individuals have many tissue samples, some individuals few, some tissues have many samples and some fewer. So it has a lot of irregularities in the structure, but and in there, basically the consortium identified all sorts of cis-eQTLs and splicing QTLs that they could find, and a whole lot more that has been published on a series of papers. I'm not going to go through it. Just going briefly over the data. So it's that. On top of that, there was the extended GTEx that basically included some proteome data and some epigenomic data for a subset of these samples. Now, GTEx finished, I guess, late 2020 officially. So after that, there's been some follow-up of the developmental GTEx that got funded. And so that is going to continue on the effort of GTEx with basically 120 donors. So it's a significantly smaller study. It's going to eventually plan to have 3,500 RNA-seq samples. And it's going to be all from pediatric cases. So this is going to be all younger pre-18 individuals, so that you could capture that sort of differences. By the way, I'm not involved in this study. I'm just like describing it. So, and then finally, there is the counterfeit from me in like a Palmer's lab that is done by Dan, that is here, is that we did the same type of thing in rats that are used a lot for psychiatric phenotypes, and something that I thought it would be good pointing out here is that basically we could use, and that we would be basically Dan, use all the visualization and data sharing tools that was developed by GTEx to present all this data so that it would be user friendly. So that's kind of a good thing about what was done with GTEx, and it's basically, so if you have your favorite animals, you could also use the tools made by GTEx. All right, cool. So how would you make your own EQTLs at home? So this is going to be your favorite Thanksgiving EQTL recipe. So what you would want to do if you want to do a QTL analysis is some sort of phenotype that you could collect in about 80 to 100 people, depending on what kind of amount of things you want to test. But generally, lower than that, it's really tough to find stuff. And so RNA-seq is obviously the most common one, but pretty much any other molecular phenotype you could measure in that kind of order. You could use it for this kind of analysis. And then you would want to get genotypes with some sort of, for some reasonably common SNPs. So the rare and singletons are not that important here because you cannot find anything for them anyways. And so that whole genome sequencing would be obviously perfect, but genotyping arrays or some sort of low coverage sequencing plus imputation, that would work too. That was, for example, what A. Palmer did in the collaboration we had for rats, that they didn't have a good genotyping array. So, and then you would want to QC and normalize and log transfer your data, so to just get rid of like super outliers, and then basically you would want to identify the confounders that you have, because gene expression is mainly driven by all these other things that you're not interested, not genetics, and that's how it should be. And so you would want to get rid of those to basically retain power for identifying genetic effects. So that's age, sex, ancestry, platform, batch, whatever you think is relevant, and not all of these are always relevant. Some of them sometimes you specifically want to exclude or not. And then you would want to pick up just general sorts of patterns that you see across your data set. So that sort of thing you could get with PCA or PEER is pretty common to do that. And then basically when you have all of those, you would set up a linear regression. You do that sort of 3 groups regression that I showed before, and then basically account for all these confounder effects, whether during that regression, or if for whatever reason you prefer you need to do it before, that's probably would work too. And then testing and so on. So now these are obviously implemented. There are a million eQTL callers out there. My current favorite is TensorQTL. That was written by, I don't remember whom, but that's it. Francois is one of them. But this was basically done for the last version of GText because we wanted to identify all the eQTLs and not just the first one. So to do that, there is this clunky procedure of one by one finding them and removing them. And going back and forth to in a greedy fashion to kind of identify all the conditionally independent eQTL signals, and that's tough. So now TensorQTL is kind of a GPU implementation that does that, and that runs on the entire GText pretty quickly. So that's something that is helpful to use even if your data set is small, so that you could get not only just the top eQTLs but also all the conditionally independent ones. So. All right, so now we have the eQTLs. How do we quantify the regulatory effect size? So this is the idea of cis-regulation. So you have two basically copies of your DNA, and then there is some sort of position that is, let's say, binding to some sort of regulatory factor, and then basically, depending on what type of genetic variation you have at that point, maybe you will produce more or less expression. So basically here I'm showing that basically you're producing a little bit more expression from that red allele and then a little less from that white, blue allele. And then basically the total expression that you're producing is going to be the sum of these two allelic expression or allele-specific expressions. Okay, so that's allele-specific expression is just the amount of allele that is coming from, the amount of expression that is coming from one of your alleles. All right, so now if you have that sort of thing, basically the regulatory effect size associated with that position is going to be, or you could define it simply as the log-fold ratio between these two things. So now this is basically the same as when you do log-fold change in your gene expression, except that this is for the two alleles. So you're kind of doing it at within one gene. And now that we are at it, I'm going to add a couple other names here because it just makes it easier. So then there is the reference or alternative allele ratio, which is basically the fraction of your gene expression for a given gene that is coming from one of those two alleles. Okay, so it's simply just, let's say, E0 divided by the sum of them. And so one convenient thing that comes out of when you define your effect sizes by the AFC is that now you could simply go back and forth between your regulatory effect size and the allele ratio that you have with the logit and logistic transformation. So basically your effect size is logit of your reference ratio or alternative ratio, and basically other way around goes via logistic function. So now a little bit more about the log AFC. So the reason we defined it this way is like you have evidence, you could actually see that that this sort of multiplicative model is actually what is happening in reality for the most part. And the other thing that is cool about it is that it's biologically very simple. So if you want to compare these sort of regulatory effects into some other assay that is fundamentally different, whether it's maybe like the SDRs that Melissa was talking about, or maybe you're comparing it to an entirely different assay that is, its readout is maybe PCR, you could actually, you know exactly what this quality means biologically. But from a mathematical point of view, this is also kind of a convenient notation to use, because basically you will have good properties for it to do consistent math for downstream analysis. So this is basically, you would have zero effect size between identical alleles. If you flip your alleles, your sign changes, but not the size of it. You could show that this is additive when you have multiple alleles or multiple variants. So it easily generalizes to cases of kind of variants that are multi-allelic. So like structural variants in SGRs, or basically when you're working in other organisms, when you have several different kinds of haplotypes available, and And so you could easily incorporate those. And then basically, if you use the absolute value of this log AFC, it's a metric, which is a very convenient thing for a lot of analysis. So, all right, so good for you. Now, how do we actually measure allele-specific expression in reality? So in reality, the way it's measured is this. So basically, you have different people that they have this sort of regulatory variation going on in them. At a given gene, this is what's happening. So you have some sort of regulatory variant that is changing the expression on two haplotypes. You have a gene, you get lucky, there is a heterozygous variant happens to be in the transcribed region of that gene. Now, when you go to your RNA sequencing reads, you can look at all the reads that align to that location and basically separate them for the two haplotypes based on that specific heterozygous SNP that you got lucky and had there, okay? Now, if you do that then for that one gene, you will get one dot like here, and then basically this dot would sit right in the middle if your two alleles are in full balance, and it will fall off the diagonal in either direction if you have allelic imbalance, or basically meaning that there is one of your haplotypes that has significantly different gene expression. Now, if you do this thing for every gene in one individual, you're going to get a cone shape like this, where each dot is a gene or a variant in a gene, and basically most of them fall on the x equals to y as expected, and then there's some sort of variation around it. And this variation around it, part of it is the sampling problems. As you see, it increases as you go lower coverage levels, and then some of it is real or artificial variation that comes from your pipeline. So we talk a little bit more about this, but if you do this right and with proper QCs, it basically gives you the cleanest data set that you've ever seen. And to me, it was always the thing that you always have these like models of your distributions that you have, and when you actually go look at the real data, you realize that it really doesn't look anything like it. And what AC data is like when you actually make it with a perfect quality, it looks exactly like the distribution assumptions that you're using, which makes it really nice. But also from a biological standpoint, it has a very high heritability. So it has 85% heritability, which makes it really good to actually pick up on genetic signals. So now, before I go forward, please stop me in the middle.
Yeah, so
all right.
in the previous slides, do you use the haplotype information?
So this one is just general. You could, and I'm going to talk about it.
Okay.
Yeah. Yeah.
So just a naive question. You said you have to get lucky for the gene to have the SNPs to be able to pick it up. Is that something that happens often enough, or it's like really you have to get lucky?
Yeah. So depends. Yes. So you have. So let's say in an individual, if you have. It's probably I should have known an exact number to just throw it at you like that, but I don't. But I can tell you, let's say, for example, in a well-covered sample, you could get probably 10,000 genes with ASC. Now, this is not like distributed uniformly. In fact, a lot of super strict restricted genes that you really love to see regulatory problems in them. They don't have ASC because they're so intolerant, they just don't have any variation in them. So the more you go in the intolerant part of the genome, the less likely it is to see that. But generally, synonymous mutations, you could have them. Or synonymous variants, you could have them. Yeah. All right. Okay. So how to make your own AC data at home. All right. So basically, to make your own AC data at home, you need one RNA-seq sample at least. So this is not some sort of thing that needs a population necessarily. And because it has an extremely high heritability, even one single sample is informative. So now you would want to get genotype for this samples. Now, if you have whole genome sequencing data, that will be perfect. Whole exome sequencing data would be pretty much as good as whole genome sequencing, because really for doing the ASC part, all you need is coding genes, coding SNPs. Now, you could do genotyping by array, but it will have downstream problems. I'll talk about it, or low coverage sequencing. And then if you really just don't have access to it, or maybe you're like downloading some data from somewhere, you could even do genotyping from RNA-seq. But then considering that you're basically then your genotyping and your analysis is not going to be independent. So you in theory can do it, but you really exactly know what you're doing so that the stuff you're doing is valid. When you have that, basically you can just throw this at the ASC read counter from GATK, and that would basically give you the ASC calculated at every ASC SNP that is available in your data. And so now, alternatively, you could phase your genome and then use this tool phaser to basically aggregate the ASC signal along a gene when you do have multiple SNPs available. So I'm going to talk a little bit more about this. So what phaser does is this is from Tully's lab. Basically, it gets a population phase data set, population phase VCF file for your samples, and then augments that with readback phasing that can do with the RNA-seq data. So for example, you could see here, if you have some sort of heterozygous SNPs that happens to be there, and you have RNA-seq data that happens to cover 2 heterozygous SNPs, then it gives you evidence to actually also phase those things are physically sitting on the same copy of the DNA. Now, this could be helpful if you're looking at very rare variants that are sitting in the coding part of the genome to get them phased. Now this is not super special about phaser. There are like a series of different tools that do this kind of thing significantly faster. And I put, for example, WhatsApp here, not because it's my favorite, just as an example, but there are others. And but one thing that phaser does that is special about it is that it actually accounts for uses that to calculate haplotype aggregated ASC data. So basically, if you have a case like this where your gene has multiple SNPs, it first phases them together and then sums the signal over that whole haplotype, accounting for double countings and usual QC problems that you need to account for. So then basically, so this is pretty nice. And it basically produces this, what you could think of 2 gene expression files. So basically, there's 2 expressions that are, one is associated with one haplotype and one is the other haplotype, and the haplotype associations are arbitrary. And so this is nice because you don't have to deal with the variance. It's easy, and it's also shareable. So for example, the entire haplotype level ASC data from GTEx is now downloadable on the GTEx portal. It doesn't need dbGaP or anything, as opposed to the actual ASC data that you need to go through dbGaP because it has all the genotype data in it. So now another thing that phaser can help you do is that you can also just give it a list of non-coding SNPs that you're interested to find what is the ASC signal associated with them. So the common example would be EQTLs. Let's say you have an EQTL, you want to know if that EQTL has a cis signal associated with it. And so you just give that to it. It basically leverages the phasing and produces that for you. Now, this seems like a simple idea, but it can get pretty finicky. So that does that. Now, the downside of it is that you lose any sort of subgene resolution. If that's something you're interested in, you lose it. But at the gene level, it actually improves both the number of positions that you have, ASC data, that you could do anything. It improves the correlation with the rest of the data sets that you have. So it's a pretty good thing. Now, I believe I'm talking too much, so I'm going to try to speed up twice as this. So maybe I'll just go a little faster. Okay. So things that can go wrong in ASC data. So this is like a good, healthy ASC data. That's how it looks like. And basically, it's a cone. It got some stuff off of it, and it got some stuff on the X and Y. These guys are positions that you either have genotyping errors or you have monallelic expression. That happens to all of us. And then there is, on this side, you have stuff that for whatever reason fall out of that code. That's chill. Now, what's not so chill is basically when you get contamination from other samples And that starts making this thing fatter and fatter. And that's the case where you have some sort of contaminating RNA coming in from another sample. So basically a part of your reads have entirely different genotypes, and basically that messes up your AAC estimation. And how you see it is that this cone starts to get wider. And then if you actually go so far that your entire RNA and DNA sample do not match each other, and believe me, that happens very not so rare, you still see a nice looking pattern, but you have thousands of positions that fall in these two areas. And that's because basically you're looking at the allele specific expression from, let's say, my genotype that is actually heterozygous at the position, but the RNA is from Jason that is homozygous at that position. So you say, oh, all of the gene expression is coming only from one side. Okay, so how do you deal with this type of stuff? You could usually look at them and get rid of them if you have too many monoallelic sites, unless you're doing something that you expect that to be the case. It's probably the problem. Now, and you could do it systematically. So this is like a undergrad in my lab, Eric, that has been working on that to basically make something with a little mixture model that can kind of capture the general patterns of OKAC and get rid of them. Don't have a name for it, but I thought we put it dumb ASC or whatever. So that's the first thing. And it's like, now the second thing that can go wrong is that basically you could have global bias, and you will have global bias towards reference alleles that you're looking at. And this is because basically we're using a single genome, it's a reference, and by definition when you're doing ASC you have at least one variant, one like a heterozygous position there, and probably by LD you maybe have more. And so as a result of it, your alternative allele always is less likely to align somewhere. Now you could mitigate it, but it's there. And so another thing that can go horribly wrong is that sometimes that one single thing can cause your read to entirely align on 20 other parts of the genome, and its expression just dies out. Now, and that's the sort of thing you could see out here. Some things go wildly out there, and overall your distribution is a little dipping down. Now, there are different ways people have tried to remedy this thing. You can't fully get away from it, but basically there are tools to help with that. So you could align to personalized genome, you could do 2-pass alignment with STAR. There's a tool called WASP that basically takes the reads, switch this position, and realigns them. And then you could also use blacklisting to basically get rid of the sites that you know aligners just don't do a good job at them. So each of these things help with certain things. So for example, WASP helps with these sort of wild cases out there, but actually in my experience, it actually a little bit increases the dispersion in your distribution because basically it just produces some randomness and gets rid of your data. But it's actually very helpful for getting rid of these wild cards out there. And same thing goes with the blacklisting. So all of them help. Depending on your case, this might not be at all a problem, or it might completely break the entire operation. So, and the final thing is this one, and this is like the biggest problem that I always see people doing, is that when you're measuring ASC data and what you're actually measuring is the gene expression associated with the SNP. It's an ASC SNP, it's a coding SNP that is sitting here. But what you're actually caring about most of the time is an entirely different SNP that is sitting somewhere in the non-coding genome, which is like interacting with this SNP in some sort of LD pattern, I mean statistically. And the phasing is not random. So sometimes they're like on the same phase, sometimes they're not, but it's not fully random. It's not like, and what happens is that basically what you would do is that you interpret your ASC data as if your ASC data is actually giving you a signal about the gene, about the SNP you're actually interested in. And so that is something that most regularly happen. You see papers that are published that are entirely doing that. So don't do it. The remedy for it is just think about it. Just know that those things are different. Now, if your case of question is simple, you could use something like Phaser that basically gets rid of that. It harmonizes your ASC and your genotype data, and then you just don't have to think about it, although your phasing is never perfect. And then, or you would use some sort of distance function that is agnostic to direction. You will lose some power because you're losing the direction, but you don't have to do that sort of stuff. All right, Now, how do I model ASC data? Binomial. It's like good old binomial, works pretty well. It actually models technical variation pretty well. Depending on your operation, this might be good enough. Beta binomial is an easy next way to basically over disperse your binomial. So if you want to capture some sort of extra binomial variation in your ASC data, or beta binomial does the job pretty well. Now, another distribution is binomial logic normal, which probably would have been much easier for everyone if it was named logic normal binomial, but that's how it is. And so what binomial logic normal is this distribution that emerges from the definition of AFC. So basically, if you calculate under the AFC framework the expected allelic imbalance for a given genotype based on all the SNPs that is carrying, basically you could show that reference ratio boils down to a normal distribution with a mean of the allelic effects of everything you don't know, plus those that you specifically do know, plus the variance of AFC of all the things you don't know. Okay? So, and if generally you don't know anything, it's just a normal distribution with a zero mean. And so the thing that is cool about it is that for a lot of statistical reasons, it's not that different from beta binomial. But if you're actually interested in interpreting the values that you're getting out of that, that actually gives you values that are in the same unit as you're doing everything else. So this is still the units of these parameters for your distribution are still log fold chains. All right, now, so this is the example of using this thing. So this is like the work we did a few years ago that we basically use this model to try to estimate the overall level of regulatory variation in a population and use that to test when you have individuals with rare diseases if they have some sort of gene that seems to have excessive allelic imbalance that is indicative of some sort of large effect mutation. And so I'm not going to go into the details, but basically so this is how this would compare, let's say. So if you do a binomial, you basically identify everything that is statistically imbalanced, but then you have so many hits you don't know what to do with it. If you do just the standard beta binomial, what you do is that you capture stuff that are clearly outside of the cloud, but basically what those things are generally crazy genes that either you're just terrible at them or they just wiggle around. That's what they do. And so in this case, that green guy is the actual true hit, and you miss that because it's not that far from the pack. And here, if you actually could model the variation you expect in each gene, then you would just pick a few genes that actually include what you're interested in. So now I'm not going to go into the detail because everyone has watched my talk. And so this is a photo of my talk in CGSI in 2019 that everyone actually watched, and it has had a pretty strong following, I noticed, with 184 views in the alternative winners league of CGSI, leading Ryan and Jason, and who talked for 2 hours. So I'm going to talk for 2 hours. All right, so let's see. I have to go a little very quickly. I think actually, yeah, dear Lord. Okay, cool. So how do we measure that? Can we measure the same effect sizes in EQTLs? Yes, we can. You could basically write the expression that you expect here in the EQTL data simply by summing up the expressions you see in different alleles. And basically, you could show with the primary school math that basically this would amount to 2 times the slope divided by the intercept plus 1. That would basically give you the same exact number for the effect size from a standard linear regression. Now, ideally, you would want to do it in a log transformed way, and that we have a tool that does that, basically accounting for the noise structure. Now, the thing is that now if you calculate the AFC from the ASC and EQTL independently, they're very consistent, so that's cool, right? They file on X and Y. But one thing that is cool about it is that ASC and EQTL, they have fundamentally different noise structures for the most part. So one thing that you could do is that you could use this consistency that you get in this scatter plots to basically tune your EQTL pipelines. Because when you're doing EQTLs, you don't have that much of a hard ground to figure out how to do things. And then there are so many arbitrary choices you have to make, like, oh, how many pairs are correct for? Do I do correct for this, for that? That helps you with that stuff. All right, so some assorted facts about GTEx. So if you look at the effects out of the EQTLs, you will see that, so here I've sorted these guys by Arbus. So on that side, you have, on this side, you have the most intolerant genes, and on this side, you have the most tolerant genes. As you would see, that the more tolerant genes have larger EQTLs, and the less tolerant one have lower EQTLs. So that's expected, but also this has ramifications. So this is important because basically at some point, if you're interested in studying it, you kind of are mostly interested in disease-causing genes, and disease-causing genes tend to go that way than this way. And so this is so. And then, but besides that, there is also a ton of correlations that are in there that if you ignore them, you can find entirely very exciting wrong findings in your papers. So for example, your effect sizes are correlated with, I don't know, length of the genes and their expression levels and how many tissues are expressed. And some of them are trivial, some of them are non-trivial, some of them have something to do with power, some of them have random reasons, like that's how evolution worked. And so you want to have an eye on these type of correlations. So another one of them is the power issue that I just mentioned. So for example, if you look at GTEx, so this is the quantiles of power. So the red is the 50 percentile, and that's the 90 percentile of the effect sizes in different tissues. Now, I have not labeled these guys, but actually those tissues with highest regulatory effect sizes are brain. And that's not because brain genes are really good at like doing variation. In fact, they're pretty constrained genes, but GTEx just has small brain samples, so everything you find in your EQTL data is large effect by definition. So that's something to have an eye. And no, another interesting fact is that if you look at the last version of GTEx, about 22% of your EQTLs have effect sizes that are larger than twofold. Now, to put that in context, that's interesting. If you have an EQTL that has that changes your expression at twofold, if you have that EQTL in homozygous, that changes your dosage more than a heterozygous loss of function would do. So that's kind of something to have. It's a good thing, a good way to think about it. So, anyone gonna keep me up? Okay. So I'm gonna quickly go over independent EQTL. So in GTEx, because we had a sample size, we could actually map a lot of independent EQTL. So this is all the tissues sorted by sample size. For the larger tissues, on average, you have 2 EQTLs per gene, and some genes have up to like 15, 16 EQTLs. So the question we had was like, so are we figuring out the human gene regulation? And we could test that. So if you actually look at the effect size of the EQTLs we have and see how good we describe the gene expression, you see this sort of improvement. So this is for genes that they have 5 EQTLs. And this is work by Nava, who is also here. You can ask her. But you have a little bit of a diminishing returns. And now if you look at the ASC data, you see that actually when you start adding more and more EQTLs, you lose accuracy. And so that sucks. And so we were thinking, why is that the case? And there are a range of reasons that can happen. And originally, when we made the AFC, we just made it for a 1 gene, 1 EQTL setup because that was what was available, and it was a pain to extend it like from logistically. At any rate, there are several reasons that that model fails when you have multiple EQTLs. So we basically made a second model that can basically calculate the effect sizes for all the SNPs that are associated with expression at once. And basically now if you calculate, you see the blue lines that you see, like now you're actually starting to describe more and more amount of variation from ASC and gene expression. And if you do this now for all the genes with all the EQTLs we know, on the blue we have the multivariant version of AFC that we call AFCN, and this would be the AFC that basically stays behind. So that's nice. This is kind of, it's not yet out, but I believe we have the effect sizes out, or if someone needs them. So I'm gonna skip over this one. So we use the AFCN to also predict the gene expression. We were just curious how this fares against elasticins and SUSY. So this is And Nick helped us with doing that. And so here I have like a significance line, but basically anyone who has done this sort of experiments, you know, there are too many moving things. But no matter how you cut it, basically this performs at a similar level to the methods that are used for TWAS. And actually the genes that come out of it, basically, so we have around 5,000 genes that are shared by both models. Those are the ones that actually have an R-square, and the ones that are not shared by models, they really don't have actually R-squared in either of them. So, and the thing that is good about AFCN as compared to the standard models that are used for TWAS is that it can basically predict the allele-specific effects on the gene expression. Now the downside of it is that it basically cannot work with summary stats, because summary stats do not have that kind of information. And there have been efforts like, so Haki's lab did an approximation of that to kind of use this model for a range of tasks, and it helps, but basically some of the major key components cannot just be transferred when you do that. And I'm not showing it here, but basically all three methods suffer the same when you have underrepresented populations. And we tried quite hard to kind of get our model to basically work, correcting for LD patterns and yada, yada, yada. We couldn't get it, at least on GTEx data, because the samples are just like, just way too low. So, so another question that we were interested in was that basically, okay, so how far have we gotten in capturing regulatory landscape of human genome at the individual level? Because that's basically the final thing that we're interested in. I want to get genome of someone and know exactly what's happening on each of those haplotypes. So, so in our model, it's straightforward to do that, right? You have, this is the genome, you have a bunch of positions that regulate it, and some of them you have figured out by AQTL, some of them you've not. You could use those, you know, to predict ASC. And your ASC is actually showing the net effect of everything, whether you know it or not. So you could use that to basically measure your gap. Now, it's a little finicky. You need to account for power and a bunch of stuff. So we did that. And this is what comes out of it, basically. So this is all the GTEx tissues sorted by their sample size. And so the total height is the amount of genes that we had some sort of allelic imbalance in them that was at least half a fold off from what we expect it to be, which is 50-50 in this case. And so this red area in here is the subset of those that we could describe with the entire available EQTL data from GTEx. Now, so the first thing is that that's an unremarkable part of it. So that's that, and that just comes to about 22.6% of everything we couldn't describe before GTEx. And by before GTEx, I mean we assumed we know nothing, not that we used other studies. After GTEx, we could describe 22% of those. And one thing that you could also see here is that the size of that red, which basically represents the knowledge gain from this entire GTEx exercise, it increases as you increase your sample sizes, which is obvious. Now, and so this is what we're showing here. So this is like the gain versus the sample size. And actually, then if you look at the knowledge gain versus the enrichment you get for rare variants in genes that are still outlier after correcting for known EQTLs, you see that you get more and more enrichment for rare variants being close to those genes. So we are capturing the signal. So that's cool. Now, the thing that was less obvious to us was this thing, that basically the remaining knowledge gap, so the things we still don't know, basically the size of that blue bars does not at all sort by the sample size. So it's not that actually because we sampled muscle with about 900 individuals while we did the brain with like 200, we actually are describing more in that. And the reason behind it is that basically if you quantify the overall level of heritable variation that is in there, which you could do, I didn't go through it, but you could do it from AAC data and EQTL data, you could see that that is actually what derives the amount of things that are remaining to be found, which is kind of suggests that basically when you're doing a EQTL study, you might also have an eye on the amount of variation that is in that sample, and that's going to be important for what you're going to capture. So this is probably going to be, I guess, my last slide, or if not one to last. And so I really tried to improve the quality of this, and I have to tell Nava sent these things properly to me this morning, but clearly didn't. Like I didn't, she did. Anyway, so one thing we want to look was like, how do we perform across ancestry? So in GTEx, there is African American, European, the rest of the answers are really, really limited. So we perform worse. That's not a surprise. It's shown in a million different contexts. And the thing that is kind of obvious, so here the darker ones are the European Americans, the next one is the African Americans, and on the red parts on the top, you show the gain. And as you see, the gain in the African Americans are significantly less, and that's just because basically there were less African Americans included in the sample, so you basically kind of did not learn really much about them. And so that's understood. The thing that I found interesting was the fact that even if you redo GTEx, so we downsampled and unified the sample sizes for both ancestry groups, you still underperform in the African population. And we could not fully pin down why is that. But basically some of the things that contribute to it is the fact that basically you just generally have more alignment issues. And then the other part in basically populations that are not just fitting to the reference panels you're using, but the other part of it is that there is just systematically higher diversity to start with in the African American population. So even though you would use the same amount of samples, you can't get the same accuracy out of it. So now this was not something we could perfectly pin it down the same with the tissues, because in the tissues you're looking at the same individuals and same genotypes. So certain things are much easier to be done. But so that's another thing. And with that, I'm going to come to my take-home message of, so I hope that this little tutorial could give you some idea about some of the nitty-gritty details of some of the genomic analysis that we do for transcriptome regulation, and maybe you could make your own favorite E-ketone ASC recipe for the next family dinner. And basically, I hope you got an idea of some of the empirical issues that you need to think about when you're using this analysis for your own applications, and basically got an idea of some of the use cases of these things. And with that, I would like to thank everyone who helped doing all of these, my team, my collaborators, funders, everyone, and you for listening to me talking so fast and ranting. Thank you.